So we just mentioned the fact that there are these things called transport proteins that exist in our biological membrane. And remember that those exist throughout the entirety of the membrane. So they extend throughout the entire thickness of the membrane because they're going to help us to shuttle things across. Well, those are going to become important in this process that we call facilitated diffusion. And this is where we have transport proteins that speed the movement of different types of molecules across our plasma membrane. And there's two basic types of transport proteins I want you to be familiar with. And the first ones are called channel proteins. And channel proteins really kind of just remind me of a tunnel. They basically provide a corridor that's going to allow for specific molecules or ions or sugars to cross the membrane. And they look basically just like a hollow. So here's an example of a channel protein. As you can see, different substances can be shuttled across that transport protein. Okay, specifically we're talking about a channel protein, and it's just kind of a hollowed out tube. Now, a second example of a transport protein is a carrier protein, and these are a little bit more advanced and they're pretty cool. But carrier proteins actually have the ability to undergo kind of a subtle change in shape that allows them to translocate very specific substances across the membrane. And we'll look at a picture to kind of describe that and visualize that a little bit better. So here's an example of a carrier protein, and what you can see is that initially, when it's wanting to collect substances from outside of our cell, it's open on top, and it's very specific in the shape of its receptor kind of region. So you can see here, it has kind of a circular divot in it that's going to allow it to collect solutes that are similar into that shape. So, for example, if we had solutes outside of our cell that were shaped like a square, that carrier protein would not be able to collect those as easily as it does these circle solutes. So it's able to collect very specific solutes and then change shape slightly so that it can spit it out inside the cell. Now, in terms of transport, we have different types of transport that are going to enable us to shuttle these substances across our membrane. And the first one I want to mention is called active transport. We mentioned facilitated diffusion, and that was the movement of substances across our membrane with the help of those carrier and those channel proteins. When we talk about this active transport is still going to be moving substances across the membrane. The only difference is that we're now moving substances against their concentration gradient. So earlier when we were talking about diffusion and when we were talking about osmosis, we were talking about the movement of substances down their concentration gradient. So we started in a region where there was higher concentration, and it kind of gravitated to a region where there was a lower concentration. So it went down its concentration gradient. In active transport, we're going to do the opposite. We're going to be moving solutes to a region where there's a higher concentration of them already, which naturally solutes and substances just don't want to do that because it's not allowing them to equalize. But if a cell needs more, it's going to have to utilize this method called active transport. And the way it does that is it uses energy. It's going to require energy to push those solutes against their concentration gradient. So it says here, active transport requires energy, typically in the form of ATP. And this slide gets really confusing, and I really don't even need you to get hung up on it all that much. It's just there more for a visual, but it's called the sodium-potassium pump. And again, it's just an example of active transport, having to shuttle substances across their across their concentration gradient. And it does that with the use of energy or with the use of ATP. So what do I want you to grasp? Grasp that during active transport, substances are moved against their concentration gradient, and it requires energy. So let's review kind of all those things we just talked about. Let's start on the left-hand side with this picture. It says we have diffusion. So we're moving substances across our membrane down their concentration gradient. And it's called diffusion, so it can be any substance at all. Now, just as a reminder, if we're talking about osmosis, that would specifically be talking about water molecules. Now let's move on over in our image, and we see this example called facilitated diffusion. So I just want to remind you that facilitated diffusion is when we're moving substances, again, down their concentration gradient, but we're doing that with the use of these transport proteins. And the two transport proteins we talked about are carrier and channel proteins. And then finally, the last type of transport we mentioned is called active transport. And this is where, again, we're using a protein to help us do this, but we're moving substances against their concentration gradient. And the only way we can get that to work is with the use of energy. Now, sometimes we get this thing called cotransport, and cotransport can be kind of confusing, but let's try to break it down a little bit. It says cotransport occurs when active transport of a specific solute indirectly drives the active transport of another solute. So if we're using energy to transport some molecule, indirectly, that transport is going to also drive the transport of another molecule. And we'll look at a picture to help kind of clarify this a little bit. But cotransport is active transport driven by a concentration gradient. So it's because of that concentration gradient that we're able to uptake some other molecule in the process. So here's an example of a hydrogen gradient. So what we see is we have, inside the cell, we have one hydrogen, and outside we have a bunch. And for some reason that cell's wanting to get rid of those hydrogens, but it's having to push them against its concentration gradient. So it's going to use ATP, or it's going to use energy to pump that hydrogen outside of the cell. Well, what happens is that hydrogen is naturally going to want to enter back in, right? It's going to want to equalize. So it's going to go through another protein, but because of that kind of gradient that's occurring, it's going to suck in sucrose, which is a type of carbohydrate in the process. So really just understand that cotransport is when you have active transport that's indirectly driving the uptake of another substance. Now, bulk transport is kind of interesting, and there's two main types of bulk transport I want you to be familiar with that can occur across the plasma membrane. And these occur by exocytosis and endocytosis. So, like we said, large proteins exist within the membrane, and large membranes can actually cross other proteins within that membrane. And let's figure out how that happens. So it says in exocytosis, transport vesicles are going to migrate to our plasma membrane. They're going to fuse with it, and they're going to release their contents inside the cell. So in exocytosis, we're going to have some little vesicle cruise on up to our cell. It's going to latch on, and it's going to dump its contents inside the cell. In endocytosis, really what happens here is the cell takes in macromolecules by forming new vesicles from the plasma membrane itself. So we're going to look at some specific pictures. Again, I like pictures to help clarify. So here we go. We're talking about endocytosis here. And the first example they're talking about is something called phagocytosis. And we've already talked about this a little bit. But it says in phagocytosis, the cell is engulfing a particle by wrapping these things they're calling pseudopodium around it and packaging it with a kind of like a membrane, a sac. But really, what do I want you to understand? Here in endocytosis, I want you to understand that our plasma membrane is kind of creating this vacuole that's going to bring in this food, right? If you remember when we talked about a tour of the cell and in phagocytosis, we said that typically we're bringing in food or digesting food with this process. So over time, once it's gathered up some food, that vesicle will pinch off, and it will move into the cell where it can then be digested by those lysosomes we've talked about before. And phagocytosis is often referred to as cellular eating. Now below that we have a process called pinocytosis. And this is where, again, we get that kind of vesicle that's formed, but it's just going to gulp in just random substances, create a new little vesicle that's going to pinch off, and then lysosomes again will come along and kind of gobble it up and distribute those contents to the rest of the cell if they need them. And pinocytosis is very nonspecific. It's just kind of bringing in anything from the outside, and they refer to this as cellular drinking. Now finally, the third type of endocytosis I'd like for you to be familiar with is receptor-mediated endocytosis. Again, this is a kind of a really fancy name for something that is just kind of cool. So look, again, we're getting this vessel that's forming with our plasma membrane. And there are these little red Ys. Well, those are the receptors that they're talking about. Receptor-mediated endocytosis. Well, in this process, we're looking for a very specific solute that we want to bring into our cell. And if our little Ys are the receptors, that means we're probably going to want to grab onto something that's maybe a triangle shape. And as you can see, we have these little purple triangles floating around. They're going to fit just perfectly on our red Ys. So those red Ys are going to grab our purple triangles. Eventually that vesicle is going to pinch off and go into our cell where lysosomes are going to be able to come in, gobble it up, and the cell is going to get whatever it needs from those little purple triangles.